I am Shanika Gajanayake, a postgraduate student in plant molecular biology at the Postgraduate Institute of Science, University of Peradeniya. My research supervisor is Dr. Shandimari Ratna, Senior Lecturer, Faculty of Science, University of Peradeniya, and the research project is funded by the National Research Council of Sri Lanka. The abstract is based on a comparative genomic study which identifies resistance gene clusters in closely related species, namely foxtail millet, finger millet, wheat, barley, maize, sorghum, teff, and panic grass, using rice, which is closely related to all these species as a model crop. First of all, it's important to understand or get to know the terminology. We'll start with what a gene is. A gene is made of DNA and act as instructions to make proteins. Therefore, resistance genes are genes that are responsible for making proteins which govern disease resistance. A gene cluster is a set of two or more genes which are closely located and have a similar function. RGA or resistance gene analog is capable of serving as a molecular marker for the rapid identification of R genes. Genome is an organism's complete set of an organism's molecular history or genetic resources. Synteny is the conservation of blocks of order within two sets of chromosomes which have been compared. Same gene found in different genomes of closely related species are known as orthologous genes, and those species are referred to as orthologous species. Let's move on to the study, and I'll start with the objectives of the study. The study identifies syntenic orthologous R genes for rice R genes in genomes of closely related species. A literature search identified rice disease resistance QTLs. A QTL is a region of DNA associated with a particular quantitative phenotypic trait. The QTLs were used to query the rice genome browser to search for rice RGAs and the QTLs. Among the retrieved rice RGAs, RGA clusters were identified based on their location. Identified rice RGA clusters were used to perform a search in Grameen and Koje Synteny databases to identify syntenic orthologous RGA clusters in the selected species. The transcriptomic sequences of the retrieved orthologous R genes were searched in respective transcriptomic databases. Literature search identified 25 rice QTLs, most of them were located in chromosome 11. The rice QTL sequences were used to query the rice genome browser, and the search retrieved a total of 75 rice RGAs. Among them, 46 RGAs were clustered, and Table 1 contains the clustered rice resistance gene analogs. Among them, 70% of the total clustered RGAs were located in chromosome 11. Rice RGA clusters were located in chromosomes 1, 2, 4, 6, and 11. The number of RGAs in a cluster varied from 2 to 6. Most of the clusters with more than 4 RGAs were in chromosome 11. The clustered rice RGA sequences were used to query Grameen and Koje databases. Rice RGA clusters in chromosome 11 identified syntenic orthologous clusters in sorghum chromosome 5, panic grass chromosome 8, chromosome 8 of foxtail millet, chromosome 4 maize, and scaffold 223 of finger millet. The table 2 contains retrieved orthologous resistance gene clusters of the species, of the closely related crop species. The highest number of RGA orthologous clusters were identified in foxtail millet and sorghum for rice chromosome 11, syntenic to rice chromosome 11. Panic grass also retrieved a considerable number of R genes syntenic to rice chromosome 11. Rice RGA clusters in chromosome 4 and 6 also retrieved orthologous RGA clusters in pannigrass chromosome 1, foxtail millet chromosome 1, and barley chromosome 7H, finger millet scaffold 369, respectively, as you can see in table 3. Some of the species, teff and wheat, did not retrieve any orthologous RGA clusters. Foxtail millet had RGA clusters syntenic to rice RGA clusters in both chromosomes 4 and chromosome 11. Transcriptomic evidence was retrieved for almost all the orthologous RGAs, which confirms the expression of the retrieved RGA orthologs, and table 2 contains the resulted transcriptomic accession numbers for the orthologous R genes as well. Rice has proven to be an effective model crop through several previous studies. The knowledge on the rice genome has been used in comparative genomic studies on other closely related crop species. Rice has a fully annotated genome, and most of the crop species selected for this study have a partially annotated genome. In some species like teff and finger millet, the genome is not annotated at all. Many studies have revealed that plant resistance genes have a tendency to exist as clusters. The study discovered that clustering of resistant genes can be observed in many rice chromosomes, although some of the rice chromosomes contain a considerably higher number of RGA clusters, and that is same for the crop species, other crops, orthologous crop species as well. Grammain and COGE databases were used to query the syntenic orthologous R gene clusters of the crop species. These are comparative genomics databases, and they contain a vast amount of bioinformatics data and tools for the study of crop and plant genomics. This study further confirmed the suitability of rice as a model crop. RGA clustering is conserved in closely related genomes. Crop synteny is an effective approach to study the disease resistance R gene clusters of closely related species. Finally, I would like to thank you for this valuable opportunity to present my findings on comparative genomics in the Asian Conference of Science, Technology and Medicine. I consider this an honor to present my findings and to share my findings with renowned scientists from all over the world. So thank you so much.